Hello, my name is Curran Howland. This presentation is on site adaptations in Teleost and Chondrichthyes due to light attenuation. Predominantly, I will be focusing on the different adaptations of these two groups and the different zones in the ocean, the photic zone and the aphotic zone. Topics that I will be going over throughout the presentation will be the properties of water. The first part is the bending of light. Can be broken into three different actions: refraction, reflection, and scattering. Light attenuation in the color scale as it pertains to depth of water increasing. The layers of the ocean itself, the photic and aphotic zones, which the photic is thought to be with the highest productivity and the aphotic is the darkest. The types of organisms themselves, the teleost and the chondrichthyes with multiple examples of each. The site, comparison of strategies from the photic zone where there's plenty of light compared to the aphotic where there's no light and organisms have to adapt to the different lifestyle. The inner working of the eyes themselves as they perceive light, and the connection from the neutral images to the brain itself. So the first property of water that I will be talking about is the bending of light, which can be broken down to three different things, as I said before. And the first two, refraction and reflection, can both be seen as an example in the first figure above. So refraction is the light penetrates from air into the water, slowing down the particles, causing it to change direction slightly. The reflection is a special case of scattering, in which a large surface area redirects radiation in a backwards direction. So this would be the sea surface reflecting light as it gets hit by this incoming radiation of the sun. Scattering, which can be seen at the bottom, is particles disperse radiation in all directions, either forward, backward, and sideways. And you can see this as the light waves are coming from the left to the right, and they are hitting the individual little particles and then therefore refracting, bouncing off, and going in any direction. The second property of water, I will be talking ocean water, is the light attenuation. And what that is the gradual decrease in light intensity as it travels through matter, as in the ocean, water absorbs the different wavelengths of light. After a certain amount of depth, the color starts to fade out. So this color scale and wavelength can be represented as this figure to the right, which has been labeled light penetration underwater. It was broken down into two different figures. So we have on the left open ocean, which was very clear water. There's nothing in the water itself, or the coastal or clear water, but we'll be focusing on the left side. The full spectrum of light fades out around 30 meters of depth. So from the sea surface to about 30 meters down, we lose almost more than half of the light colors. Green stops around 175 meters. Blue light penetrates to a depth around 200 meters. Even these ranges can be altered by turbidity effects on light intensity. So this could be it could be affected by waves, particles in the water, or plankton blooms. But then we see that the light is the blue light stops around 200 meters. After this zone, in this zone, it is called the twilight zone, which is there is little light penetration. Below this is the aphotic zone, and there's only darkness. And then I will be showing you a video that demonstrates how color can be lost within the first 35 meters of depth.
This apple looks red because molecules in its skin interact with various wavelengths of sunlight. Now, most of these wavelengths are actually absorbed by this depth red. Now, how does it detect the reflected light in our brains? Red apple. It seems pretty simple. Well, it's not so easy to explain the colors of many things like water. So we're going to explore the question, why is the ocean blue? And to do that, we're going to run a simple experiment. All we need is this red apple. We're going to use these colored toppers, and we're going to use my dive gear. Because you see, I'm going to dive down into the depths. I'm going to see if the colors look any different underwater. What do you think is going to happen? I don't know. Let's go diving. Here we are at 1 meter. Now the apple still looks red, right? Now let's descend to the wreck at about 35 meters. The apple looks dark gray or black. Did the apple really change color? No. When I turn the camera light on, the apple is still red. And look, we can see some of the colors here on the toppers. Do you know why we can't see red, but we can still see green and blue? Well, at the surface, the apple looks red because it reflects red wavelengths. But at this depth, there doesn't appear to be any red wavelengths for the apple to reflect. And it absorbs all the other wavelengths, so it looks black. But what about the color toppers? You can see the blue and green colors are still showing up, even if they appear a little bit different. So that means that at this depth, there might not be any red wavelengths, but there's still some blue and green wavelengths. So why does this happen? It's because red wavelengths are absorbed by the water first, and blue and green wavelengths penetrate deeper.
That video just showed how the colors start to fade out as depth goes on. So, on to the next part. Layers of the ocean. As I stated before, that there's two layers we can think of. There's the photic zone, which is from the top portion, sea surface, to about 200 meters on average. It is the sunlit surface layer of the ocean down to the depth where light is just sufficient for photosynthesis. Meanwhile, the aphotic zone is part of the ocean where light does not penetrate and no photosynthesis occurs. This can be seen on the figure to the right as average depth. There are two portions within the aphotic zone. One is called the twilight zone, which is when there is little downwelling light and organisms that adapted can still see. This region can range from 200 meters to either 1,000 or 4,000, depending on location. Meanwhile, the second portion, as it is closer to the ocean floor, there is no light. The depth range can be thought as the bottom portion of the twilight zone to either 6,000 or 10,000 meters to the floor. So I'll be going to the in-depth portion of the photic and the aphotic zone after this. So as I introduced in the previous slide, it is comprised of two lesser parts, the euphotic zone and the dysphotic zone. The euphotic zone is the upper part of the ocean that receives the bright and clear sunlight. The dysphotic zone is the water layer beneath the euphotic zone, where there is dim blue light penetrates, but is not sufficient enough to sustain photosynthesis. Organisms use sight to find their food with the available light. This is why they need the bigger eyes and all the colors so that they can determine what is food and what is not. Between the ocean surface and it's about 200 meters deep. This is the first 200 meters, as stated before, with an optimal wavelength of absorbance of 500 to 530 nanometers, which is the reds and oranges that would eventually fade out. Now the aphotic zone. The portion of the ocean where there is no visible light. Organisms using chemosensory or specialized eyes to see in the low to no light. ranges between 200 to 1,000 meters deep or to the ocean floor. Wavelength of light absorbed best at is 470 to 480 nanometers. This is the blues and slight greens. Now on to the types of organisms. So we have the teleost, which are the bony fishes, and the Chondrichthyes, the sharks, rays, and skates. So I split this up into two different things. So we have the photic representatives. So I chose to use a yellowfin tuna, and for the teleost, and then a great hammerhead shark for the Chondrichthyes. And then for the aphotic zone representatives, for the teleost, I used barrel eye, and for the Chondrichthyes, the Portuguese dogfish for the aphotic zones. Each have been adapted to living in their zone in the ocean, which was split up into further. There are plenty of more species that inhabit these environments. I'll be focusing on these 4 species as they show strategies that is best exemplifies sight adaptations to decreasing light. So now we move on to sight. There are 3 main functions of the eye. 1 is to collect the incoming light. Secondly, focus images on the retina. And then the third, transforming the images into neutral signals. If the organism does not know the individual image, they can use clues from the surrounding area to relate it to something they have encountered beforehand. I will go further into the individual aspects of the most important parts of the eye, and it will pertain to both teleost and Chondrichthyes, as they are only varying slightly. So the components of the teleost eye. The eye is composed of 7 basic parts, as these are critical for sight. I will individually explain the importance for each as they show up on the screen. The first part of the cornea, which acts as the outermost lens. Secondly, the iris, as it controls the amount of light entering the eye by adjusting the pupil. The lens focuses light on the retina. The sclera, which forms the outer layer of the eye to protect the inner structure. The choroid and tapetum lucida sometimes work together to reflect light through the retina back into the inner layer of the retina. This can be seen on many organisms, as it is characterized as a reflective shine in the eye when it comes to light. Finally, the retina. The purpose is to convert the reflected light into neutral signals and then send them to the brain. Now we're on to Teleost. Teleost is a very diverse species, as it ranges from our fast-swimming fishes, both yellowfin tuna and the swordfish, to the clownfish that calls coral reefs its homes. Diversity is thus resulting in large types of eyes, which can be seen in the pictures to the right. Along with the photic fishes, the aphotic zone has a large population also, consisting of barrel eyes, hatchetfish, and anglerfish. But no matter where the fish is located, the eyes grow throughout the entirety of the fish's life. The retinal tapetum is the retroreflector, reflecting visible light back through the retina, increasing available light to the photoreceptors. The teleost sight strategies from twilight to aphotic. This is as depth increases, their adaptations to the diminishing light has increased. So one of the strategies is the yellow intraocular filters. Their functions can be broken down into 2 parts. First, the reduction of chromatic aberrations, which a chromatic aberration is the material effect produced by the refraction of different wavelengths of electromagnetic radiation through slightly different angles, which can result in failure to focus. Secondly, a reduction of glare, thus improvement of the contrast vision, or having a transparent head with green eyes which can filter out sunlight coming from the ocean above, and then allowing the teleost to see objects above, determining if there is danger or a meal. Finally, having tubular eyes. These eyes have large lenses that can be good for have good lighting properties, and are predominantly used to look upwards from the downwelling light. The video I'm going to show you puts two of the three strategies together in one organ, the barrel eye. So this is the transparent head and the tubular eyes.
A small, dark fish with large fins, a tiny mouth, and a remarkable pair of eyes. The two green spheres in these video shots are the lenses of its tubular eyes. Behind the eyes is a set of large scales. The eyes are enclosed within a transparent shield, sort of like the canopy of a jet fighter. In front of the eyes are two dark capsules containing the fish's olfactory organs. Typically, Macropinna sits quietly in the water, using its big fins for stability, while it scans the water above for food. When it spots food, it can rotate its eyes to look forward, to include its mouth in the field of view. We speculate that Macropinna steals food from siphonophores, elongate animals with tentacles that capture prey that swims into them. And we think that Macropinna swims into the tentacles and steals the food from the siphonophores. The shield over its eyes protects those sensitive structures from the stinging cells in the siphonophore's tentacles. It can also rotate its eyes to avoid predators, or to avoid being captured by scientists. This is Bruce Robinson. Research Institute.
Now that we have talked about the teleost strategies, now we're going to talk about the components of the Chondrichthyes eyes. In comparison to the teleost, the shark's eye is very similar, but there is one major difference. This is the nictitating eyelid, which protects the eye during the attack. You can see this as it is the moving structure on the figure to the right. This protects the eye during an attack in case if the prey tries to scratch the eyes during the struggle. This is a quick video showing how useful this mechanism is. Just bear with me one moment.
Behind the retina, called the tapetum lucidum. Its purpose is to reflect light back through the retina a second time. It's very similar to the eye of a cat. If you look into a shark's eye at night with a light, it'll actually reflect back at you, just like a cat's eye. This is the low light sensitivity of a shark's eye. They can see amazingly well in light that to us would just look dark. Some estimates say that the shark's eye is 10 times more sensitive in low light than human eyes. And get this, sharks also have eyelids. They're the only fish with eyelids. They're called nictitating membranes, and they close them when they're attacking prey at close range to protect their eyes.
Quick video, and they were talking about the tapetum lucidum working in the eye to refract the incoming light for the retina. So, as talked about the very diverse species of teleost, we also have our very diverse Chondrichthyes, which are vertically distributed as the same as before as the teleost. But predominantly, most of our sharks that we all know are in the photic zone, or the euphotic zone, which consists of our very well-known great hammerhead, great white shark, and the whale shark. While the aphotic zone have the Portuguese dogfish and frilled shark. Even though they don't have as many, they still have a abundant amount of species. Okay. Chondrichthyes site strategies. So, as in the ocean, like Teleost, the eyes continue to grow throughout the lifespan of the chondrichthyes. So, in the pelagic and mesopelagic, typically they have the largest eyes, which allow the available light to penetrate, compared to the bathypelagic, which have smaller eyes but with larger pupils. This increases the visual acuity and ability to detect bioluminescence. Along with the bathypelagic, below in the aphotic zone, large eyes with immobile pupillary and low focal ratio would equate to increased detection amplification of low light levels. This allows them to see in very low light levels. They can pick out the contrasting images of other organisms or objects. Along with the adaptations of the aphotic zone, we also have bioluminescent eyes, which this video will show some strengths and weaknesses, mostly strengths of the sharks that use these in the mesopelagic area.
Sharks that live in the deep ocean have evolved to have special eyes that help them see in the low light conditions. Researchers have discovered the science behind the bioluminescent shark's eyes by comparing them with the eyes of other sharks. Their eyes allow them to see the points of light that reach the depths of the ocean and recognize other bioluminescent fish. One important discovery made by the study found that there is a space between the lens and the iris of the shark's eye that lets extra light into the retina. According to study researcher Julien Claes, a biologist at the Catholic University of Louvain in Belgium, there are about 50 different shark species that are able to produce light, about 10% of all currently known sharks. They live at depths of between 650 to 3,300 feet below the surface of the ocean in an area called the mesopelagic twilight zone. Being able to produce light helps to keep them hidden from predators by camouflaging against the sunlight above. It also helps the sharks identify potential mates and hunting partners that belong to the same species.
Similarities between Teleost and Chondrichthyes. As both Teleost and Chondrichthyes share a common ancestral heritage, they can use their ret meables. This is a structure that allows the fish to warm the areas around the eyes to increase retina rate of response to change. As you can see in the figure to the right, it is the structure that says ret, and then there is a red and then blue line. So this is a countercurrent heat exchange with warm and cold blood. Due to the countercurrent exchange of warm and cold blood, by having the warmer region behind the eye allows for the eye to receive incoming light and process it into neutral images. This can be beneficial to prey capture or evasion. And there is a quick video showing the inner workings of why it is important for countercurrent exchange.
Some animals go through a mechanism known as countercurrent heat exchange. This mechanism allows for the animals to save energy by using parallel pipes, which are the arteries and the veins, that allow blood to flow in opposite directions to and from the appendages. Here the arteries are displayed in red, and they are typically found deep within the appendage, while the veins, displayed in blue, are superficial. They connect in the middle by capillaries. For the purpose of display, the pipes seen in the outer part of the model exemplify regular blood flow which does not conserve heat. Rather, heat would be lost to the environment, thus steadily declining blood temperature. This process is held when the organism is at optimal.
Moving on. So the sight, eyes perception. Most vertebrates share the similar eye perception apart from a few extra adaptations, but in most eyes the retina is comprised of rods and cones. As you can see on the figure to the right, the left is rod, the purple structure is a rod, and then the green structure is a cone. Within each of the outer segment contains a visual or photosensitive pigment. A photosensitive pigment are unstable pigments that undergo a chemical change when they absorb light, which are then held in place by an external limiting membrane. Individually, the rod's actions are responding to the dim light, but with poor sharpness and without color perception, but does contain more visual pigments and is more sensitive, while the cones are divided into populations, and each population has a different visual pigment. Site adaptation to change in light. On the figure to the right, the figure depicts the adaptations in teleost towards light and dark. Light. When light is present, the rods are farther from the external limiting membrane and farther from the light source, and the cones are closer to the external limiting membrane and closer to the incoming light, as there is plenty of available incoming light. While when the dark adapted adaptation is that when rods are closer to the external limiting membrane and then are also closer to the light source, and then cones are farther from the light source and external limiting membrane. This allows the available downwelling light to penetrate the eye when there is little to no light at all. And finally, the connection to the brain, what makes it all work. So, as the rods and cones are connected to the brain via bipolar and ganglion cells. Axons and the cells pass along the optic nerve to the optic lobe. The video will show a more in-depth version of this, but it is for human eyes, but since all vertebrates share the similar eye structure and information, this works.
In this installment, I will discuss the retina. The retina contains the neural component of the eye. When light reaches the back of the eye, it enters the cellular layers of the retina. The cells of the retina that detect and respond to light, known as photoreceptors, are located at the very back of the retina. There are two types of photoreceptors, rods and cones. Rods allow us to see in dim light, but don't allow for the perception of color. Cones, on the other hand, allow us to perceive color under normal lighting conditions. Throughout most of the retina, rods outnumber cones. In one area called the fovea, however, there are no rods but many cones. The fovea represents the area of our retina that provides our highest acuity vision, and thus is at the center of our gaze. When light hits photoreceptors, it interacts with a molecule called photopigment, which begins a chain of events that serves to propagate the visual signal. The signal is transmitted to cells called bipolar cells, which connect photoreceptors to ganglion cells. Bipolar cells pass the signal on to ganglion cells, which leave the eye in a large cluster at an area called the optic disc. The optic disc doesn't contain any photoreceptors, and so represents an area of the retina that can't process visual information, creating a natural blind spot. However, we normally don't notice our blind spot. The brain uses information from surrounding photoreceptors and the other eye to fill in the gaps in images that are processed by the retina. After leaving the retina, the ganglion cell fibers are called the optic nerve. The optic nerve carries visual information toward the brain to be processed. There are two other cell types in the retina that should be mentioned, horizontal and amacrine cells. Horizontal cells receive input from multiple photoreceptor cells. They integrate signaling from different populations of photoreceptor cells, make adjustments to the signals that will be sent to bipolar cells, and regulate activity in photoreceptor cells themselves. Amacrine cells receive signals from bipolar cells and are involved in the regulation and integration of activity in bipolar and ganglion cells.
In conclusion, light attenuation determines the depth of the photic zone and then the aphotic zone. The decreasing available light constricts organisms to adapt to the little or no light environment. The aphotic zone starts around 200 meters deep, and then also sight has three main functions in either Teleost or Chondrichthyes. First is collecting light, second is focusing images on the retina, and then the third is transforming images into neutral signals. Also, the tapetum lucidum is a key component of the eye, as it reflects the extra light into the retina so that the rods and cones can be able to identify what's happening. And then the size of the eye is dependent on the layer in which the organism inhabits. And then retinas are made up of rods and cones, and then the connection to the brain goes from the retina to the rods and cones, the photoreceptors, bipolar cells, ganglion cells, and the optic disc, then the optic nerve, and then the brain. And these are my citations.